Blog Talk Radio.
Welcome to the AME Food Testing Show. Today's topic, unlikely contamination of produce through root uptake, with Dr. Kalima E. Kniel-Tolbert, Associate Professor, Microbial Food Safety, Assistant Chair, Department of Animal and Food Sciences, College of Agricultural and Natural Resources, Food and Waterborne Protozoa and Viruses, and use of non-thermal methods to inactivate organisms on fresh produce, University of Delaware. She received her bachelor's, master's, and Ph.D. degrees from the Department of Food Science and Technology at Virginia Polytechnic Institute and State University in Blacksburg, Virginia. She has over 50 publications. She is a very well-known author in the area of food science. Now, welcome with me, Dr. Callie Kniel.
Good morning, Andy. Thank you very much. Thank you very much for that wonderful introduction and the invitation to be here with you today. Good morning.
Well, welcome to you and your expertise on this most compelling topic. I note again, you have over 50 publications and a textbook, all on food science and food microbiology. Let's address the issue from the very basic. What is a foodborne pathogen?
Sure. These are enteric microorganisms, meaning they come from the intestine, and they're either bacteria or viruses or parasites that use food and water as their vectors to get to the human or to the animal host. Some foodborne pathogens are zoonotic, which means they can be transmitted from an animal host to a human host, and others may be only strictly able to infect humans and will be shed by humans, and then that they are deposited on or near food through contaminated handling or contaminated water or other fomites or equipment that might be used in the preparation or the growth of food.
Excellent review. Let's talk about our topic. How do generally these foodborne pathogens that we're talking about, how do they contaminate food products?
Well, there are a lot of possible routes of contamination, and I think because those possibilities are almost infinite, that's why we have such a hard time controlling them in all facets, although I would say that our food production system is one of the safest in the world, and certainly we are doing a very good job of handling them, but there's always more to learn. One way is through food handlers, and food handling contamination can certainly occur at various parts of the chain from farm to fork. Often we divide food production into two areas, pre-harvest and post-harvest. And I think we'll be talking a lot about pre-harvest today. And when we think about possible routes of contamination in the pre-harvest environment, there's improperly composted soil amendments, there's compost, dust from these sources, and air particles is certainly an area of a lot of research consideration today. There might be contaminated water sources. And of course, this contamination is intermittent, and that makes testing very difficult. It also makes it harder for us to understand maybe the root of the problem. Animal intrusion in a pre-harvest environment is a big issue. Birds, reptiles, amphibians, mammals, all of those may carry foodborne pathogens. And certainly equipment is also an issue. And then in the post-harvest environment, there are just as many possible ways of contamination, including water that's used for sanitation or water that's used to carry the food products like fresh produce in a flume, for example, or even more and more handlers that may be involved, the equipment and packaging materials. Really a complicated issue.
So the numbers of vectors that can actually contaminate the food are many. Let's chat about our main topic of the day. Can fresh produce be contaminated through the roots directly into the product?
You know, this has been a fascinating question, and I think if we put it in context following the 2006 outbreak of gastroenteritis that was associated with the pathogen E. coli 015787 in contamination of fresh spinach, there has been a great deal of attention focused just on that question and trying to understand, again, to put it in the bigger context, is where can we use good agricultural practices and how do we understand how pathogens interact with fresh-grown produce. After this outbreak, the industry recognized, the leafy green industry in particular, as well as all of that of fresh produce, recognized the importance of understanding this question: how do bacteria interact with fresh produce, such as leafy greens? And the question of how do bacteria get into or on the product was really important. And I think researchers have done a great job at looking at this question in a very systematic approach, starting back in 2006. And even before that, researchers were looking at laboratory experiments where they were growing crops, growing leafy greens, for example, hydroponic growth mostly, so just water, no soil, and they would expose the root tips of these plants to millions of bacteria, millions and millions. And that high number of bacteria under hydroponic growth was able to get up into the root and get into the leaf, into the foliar portion, the edible portion of the plant. And of course, that made a lot of people very nervous. And then scientists started to reassess the problem, because while those experiments are excellent, they're wonderfully designed and they're very informative in telling us what bacteria can do, it doesn't necessarily show us what's happening in the field under real-world situations. And so we've grown into a more real-world view of how we conduct our experiments, moving from the lab bench to greenhouses and then to the field, actually. And field trials are undergoing all over the country in different climates, which is really important, and different crops. And we're now finding that the bacteria tend to bind to the colloids, they tend to bind to the soil, and they're really not going to get into that root. And even if they do, under extreme circumstances, if there's sewage sludge or a flooding event, something like that, some bacteria may get into the root. That doesn't correlate directly with internalization into the leaf, into that edible portion. So while it may happen, it's highly unlikely that getting into the root tip is going to then result in a plant that's going to be contaminated within the leaf.
Let me ask you this question, Dr. Kniel, and it's maybe a bit of a naive question, but doesn't the plant have its own resistance mechanism to these types of bacteria entering their own internal protection systems?
You know, they do. Actually, plants are very much like humans. They have immune responses, just like we do, and they have almost a three-tiered immune response with various hormones and chemicals that are released, just like animals and humans. And it's very fascinating, and more scientists are studying this. We're actually looking at how the plant responds to different bacteria or viruses that might be applied to the leaf or to the root. And what's really interesting is that the plant knows very well how to deal with a plant pathogen. And now to kind of get back to your first question about foodborne pathogens, these are a little bit different. These are enteric organisms. They are organisms that are very happy growing within the human or an animal gut, inside the intestine. It's a different environment from the plant, from a plant surface. Plants don't seem to respond the same way to all foodborne pathogens. And that's where we really need more research. We need to have a better understanding of how do they interact on that plant surface and how does the plant respond. And there are big differences between some of the top foodborne bacterial pathogens. For example, Salmonella versus pathogenic E. coli. Salmonella may be more similar to plant pathogens. So Salmonella may actually be able to get into, on a leaf surface, more than E. coli could, for example, and they will act differently. They will respond differently, Salmonella and E. coli, to plant cues, to the sugars that are produced in the plant leaf even. And so there are some intrinsic differences in the bacteria that we as scientists need to try to figure out so then we can better understand the problem. And the way the bacteria get into the leaf surface is different from the root. They actually can go through stomata, which are tiny openings. They're sort of where the organism or the plant can breathe. And they look like little mouths. If you look at them under an electron microscope, you can see them with little openings. There are two cells that surround this little opening called a guard cell, and that helps to open and close it. And it's always functioning. These are really amazing little cells. To me, not being a plant scientist by nature and now looking at plants this way, they really are amazing. They allow gases like carbon dioxide and water vapor and oxygen to move in and out of the leaf. And while they're doing this, they may also allow movement of pathogens. And the plant pathogens work with these stomata, and they can actually trigger them to open them to get in, because that's what a plant pathogen wants to do. So now we're trying to understand, do human pathogens do the same thing? And could we then construct plants or find plants that wouldn't allow a human pathogen to do that? So there's some fascinating areas of research.
Well, that's particularly interesting as I think about the premise of today's interview is that can fresh produce be contaminated via the roots? Would a plant, particularly lettuce or fresh green leafy, be discolored or give some sort of outward notice that they're being attacked by these bacteria, or would it just look as fresh as any other?
That's a great question. The plant pathogens that I'm mentioning, the plants will respond to those, and they will turn kind of a yellowish color sometimes when they're infected. And we don't seem to see the same thing with Salmonella, although I will say that there isn't enough research that's done. We really are looking into that, and there are some great plant pathologists that are teaming up with food microbiologists and trying to understand that, because wouldn't it be great if you could tell? That would be a wonderful advantage. So that's something that's being looked at.
Well, Dr. Kniel, you have offered some fascinating historical documentation on your studies. What does the recent science suggest about how food producers, and again our audience is primarily food production, food quality, food safety, and food security managers, what does the science suggest about how we can prevent these foodborne pathogens from entering via the root of the plant?
I think it starts with good agricultural practices. We really need to maintain what growers have been doing all along and working very hard to use properly composted soil amendments, properly treated soil amendments, all of that's going to be worked into the soil, that's going to get at the roots. We need to be very conscious of the water that's being used for irrigation, if it's going to be drip or overhead. We're going to need to be very conscious as well about the different types of animals that may be getting into the crops. Certainly that's been something that's been looked at in the past. There's a lot of research now going into the buffer zones, and certainly produce growers are facing new regulations with the proposed produce rule that was recently released, which stipulates certain metrics for water and certain times for composting of soil amendments and such. I think adherence to all of those is going to be really important, followed up by sanitation. If we're going to be washing products, is that wash water being well monitored for the amount of chlorine that's being used or other sanitizer that's there? I think coupling those is going to be extremely important to try to prevent cross-contamination. You can't be very careful in the field and then have an event of cross-contamination during packaging or during the washing. And at the same time, trying to filter in some of this new science that we're learning, the new ways we're learning about how pathogens interact with the plants, and try to integrate any of that we can in the pre-harvest environment.
Well, Dr. Kniel, you've given us some excellent suggestions. We've heard them before, but the repetition is really going to help us to look at the way we grow food and the way that we can potentially improve or minimize the intrusion of these organisms into our food products. Let me ask you, what further research would enlighten producers and consumers to protect us from these foodborne pathogens?
Well, some of the research that is going on now is at a very basic level where we're trying to understand what drives a certain bacteria to want to live within a plant or live on that plant surface, that leaf surface, which is a really different environment for an enteric human pathogen. And by understanding that, we might be able to incorporate healthful bacteria or bacteria that are normally on the plant that would not allow the colonization by human enteric pathogens. Similar work has been done with poultry production, or even when you think about our own health and people wanting to eat yogurt or have probiotics to have very healthy intestines. You want to make that environment on the leaf surface an area where the pathogens won't want to survive. So that's an interesting area of research, and there's a lot of new studies that are coming out all the time on that. I think the best thing to keep in mind when it comes to produce safety is that outbreaks of illness and outbreaks where we find pathogens on these plants are still rare events to show that people are doing the right things. I think we're doing a very good job. Industry's working very hard, and certainly now working even harder with the new rules to try to reduce any type of contamination. But I think as everyone maintains their vigor and we see some of this new science that's coming out and being implemented very quickly by the industry, who's very receptive to all the research that's going on.
Dr. Kniel, I certainly appreciate your time this morning and your energy in this particularly important area for food production, food quality, food safety, and food security managers. I'd like to now allow you to conclude your remarks with a review of what are foodborne pathogens, how do they contaminate food products, and dispel the sometimes preoccupied type concern that these pathogens actually can penetrate the root system of the plant, and then just concluding remarks about the future of our research in those areas.
Well, foodborne pathogens are those enteric microorganisms that use food or water as a vector to get into their animal or human host. And while these organisms are not normally found on foliar surfaces or in the environment where we grow fresh produce, for example, they seem to be fairly well adapted to these areas. So by means of continuing good agricultural practices, by doing the best we can to try to eliminate them from contaminated water or soil amendments in particular, we can reduce the risk of those pathogens being present in soil or water that's been applied to our plants. It seems that by looking at many, many studies that have gone on, that there is, for the most part, little to no observed internalization in soil-grown plants compared to hydroponic solutions, for example. And even in those studies where internalization is observed in soil-grown crops, it's sporadic or at very low levels and does not then correlate with internalization in the leaf tissue and what we're eating. So I think that that's very good news for growers to just continue what they're doing, as well as integrate as much of the new science and new understanding of the pathogens as we try to continue to safeguard the crops that we're growing.
Dr. Kniel, that was an excellent summary of today's discussion. I'd like to keep you on tab in the event of any of your research being of direct application to our listenership, and invite you back on the show.
Thank you very much. I look forward to it and look forward to updating you on all the exciting research that's going on.
Thank you very much, Dr. Kniel, and thank you again for joining us from the University of Delaware.
Thank you. Have a great day.
Bye now.